So in the next 50 minutes, we will discuss about the current requirements of the registration in Europe of COVID-19 tests. The registration of the IVD test in Europe is done under the 98/79 directive, also called in-vitro diagnostic medical device directive. This directive, in fact, it sets out a goal for all the European countries, and each country, in fact, has to make the transposition of this directive in its own national law. Well, what does it mean? It means that depending on the country, the interpretation on how to achieve those goals may be different. So at least to be slightly different. It also means that it's possible, in fact, to have some specific country-specific requirements which are indeed due of this different interpretation of the common goal. So any manufacturer who places a device on the market has to notify its competent authorities in the country where he has place of business. When this legal manufacturer, so of course when I'm talking about the manufacturer, I'm talking about the legal manufacturer, the one who has the name on the product and is legally responsible of the product. So when a manufacturer doesn't have its place of business in Europe, which may happen for a legal manufacturer coming from China or United States, they have to designate an authorized representative in Europe. This authorized representative is also called EC rep. And the registration of the product in Europe has to be done, therefore, in the country where this authorized representative has its own place of business. Once the device is registered in the member states, the manufacturer can make the device available in the whole territory of the European community. So when I'm talking about competent authority, I'm talking about the authority which is in charge in every European country of the registration and the control of medical devices, not only in vitro diagnostic devices, but in general of medical devices. For instance, in Switzerland, the competent authority is Swissmedic, and to give you another example, in UK we have the MHPRA agency, which is called Medicine and Healthcare Products Regulatory Agency. As I told you before, because of this transposition in national law of the 98/79 directive, we have those country-specific limitations. Those country-specific limitations are general to all the in vitro diagnostic devices which are placed on the EU market. Those country-specific requirements apply to labeling. We have country-specific requirements in terms of language, which apply to labeling, instruction for use, for instance, but also to the registration of some economical operators. Some countries will ask, in fact, for the registration of importers or distributors on their with the competent authorities. Additionally to those known country-specific requirements, which, as I said, apply to all the IVD devices which are to be placed on the European market, we also have, with the COVID-19, we have specific limitations. Those specific limitations are solely applicable to the COVID-19 tests. Here you have some example of the limitation which apply specifically to these COVID-19 tests. You see here that in Belgium, in March of this year, in fact, there was a royal decree which was adopted making an interdiction, in fact, of putting into service, in fact, self-tests which are intended for COVID-19 detection. So apparently this Belgium decree will apply up to September of this year. So additionally to that, we also see that in UK currently this information, I took it last week from the MHRA website, so UK. So this is the competent authorities in UK, where it says that it's currently illegal to supply tests for use by member of public. What does it mean? Again, is that in fact you cannot supply tests for self-tests. Additionally to that, we can see that we may have, as I told you before in one of my previous slides, normally once you register your device on the European country where you legal manufacturer or the EC rep has its place of business, then you are supposed to be able to put on the service your product in all the European countries. However, this is not the case for all the European countries, depending specifically for COVID-19 tests. As you see here, the third paragraph that I put on this slide, you see that in Ireland, for instance, all the stakeholders who arrive on the market with a new IVD test for COVID have to contact the competent authorities and the competent authorities will advise if the test may be put on the market or not in Ireland. So we see that every time now with the COVID test, when you decide to put this type of test on the market, you really have to pay attention not only to the registration as a whole in Europe under the 98/79 directive, but you have also to be sure and to check, depending on the countries where you would like to make your product available, if this is possible or not. Some of our clients are asking us about the humanitarian use of non-CE mark devices. While this is a country-specific program, it means that every country has its own program and its own application. Once you apply in one country, you cannot make your product available in the other European country. It has a limited authorization in time and condition of use. And the most important thing is that it does not waive the assessment of the technical documentation by the competent authorities in the country where you apply. How does it go, the procedure for registration of an IVD medical device in Europe? Well, it goes through a process which is called Conformity Assessment. The Conformity Assessment procedure is the name given to the process that is used to demonstrate that the device meets the essential requirements. Those essential requirements are listed in the Annex 1 of the IVDD. During this conformity assessment process, in fact, we are checking that the technical documentation establish proof of performance, design control of the design and manufacturing process. We will check the quality management. It checks the quality management system of the legal manufacturer and also the post-market surveillance, which has to be set up by the legal manufacturer prior to putting on the market a new IVD device. Once the conformity assessment procedure is successful, the legal manufacturer can issue a declaration of conformity, and the device can be registered on the US market by notifying the competent authorities, again in the country where the legal manufacturer or the EC rep has its place of business. The conformity assessment, in fact, all this process where the review of the technical documentation and of the quality management system is assessed, in fact, depend on the classification of the device. The classification of in vitro diagnostic medical device under the IVDD is specified in the Annex 2 of the directive. First, we can see that the classification is based on risk. The highest risk are those devices which are classified in list A, so which are found in the list A. Then we have the list B. We have the self-test devices, which are considered as low to moderate risk. And then we have the general devices, which are devices of low risk. So to give you an example of the tests which are found in each of those lists. So we can see here that list A, we have tests which are called as being at high risk, such as the one used for blood grouping under the ABO system, and also devices which detect HIV or hepatitis. In the list B, we have what as called moderate risk devices, which are the ones that are again for blood grouping per anti-Duffy system, but we also have devices which detect toxoplasmosis. This is mainly those devices detecting toxoplasmosis are found here because an infection of toxoplasmosis in a pregnant woman will have an effect also not only on the mother but also in the offspring. Additionally to that, you can see here that we have a test for cancer, PSA, which is a marker for prostate cancer. I would like to underline here that this is the only test for cancer which is found in this list B, or none of the other cancer tests are found in list A or B. And this is mainly one of the major reasons why, in fact, the IVD directive will soon be replaced by the IVDR regulation. Well, Rima will discuss this point a bit later during this webinar. We can see also that we have here the blood glucose self-test, because blood glucose, it has an incident on the health, can have a direct incident on the health of patients. But most of the other self-tests, like pregnancy, cholesterol, so on, are found in another category, which are called self-tests. So all the other self-tests which are not for blood glucose will have their own category of self-tests. And finally, we have this general category, which is called like that, and it's said as being at low risk, which is not totally true, because you can see here that we can have in this category the instruments like the ones used in a lab for in vitro diagnostic processes, like ELISA reader, for instance. We can also have specimen receptacles, and of course we have all the other tests which are not covered by list A or B and which are not self-tests. So what would be the classification of COVID tests under the IVD then? We will have two types of classification. In fact, in one hand, we will have the self-tests, which will have the low to moderate risk. Those are to be used at home by patients themselves. And then we will have all the other COVID tests, which are intended for a health professional use, which will be found in this general category of devices. So we are strongly encouraging legal manufacturers who are having tests for intended for health professional use, who they would like to put on the market here in Europe, and also the ECREP, European representative of legal manufacturers which have their place of business outside the Europe, to make conformity assessment of their test, of the documentation available before putting the test on the market. I'm particularly underlining here the importance of this conformity assessment, particularly for the legal manufacturer who are buying their product on other markets as an OEM product, and then they are putting it on the European market under their name and responsibility. In this case here at Medide, we are proposing, in fact, to review that all the technical documentation based on the requirements of the in vitro diagnostic directive, and we checked the compliance of this of the documentation with the essential requirements which are found in the Annex 1 of the IVDD. The legal manufacturer shall hold a quality management system. The quality management system standard is the ISO 13485. This is the standard for medical devices. You need to have a proof of it, of the quality management system. The ideal proof will be a certification, ISO 13485 certification. However, I would like to underline here that certification by a notified body of your quality management system is not a legal requirement. So you may have proof of your quality management system, showing you can have a quality management manual which shows what are the processes that you implemented in your quality management system. I pay attention here that the post-market surveillance, it's a process which shall be implemented into your quality management system. So from a perspective of control of the quality management system for the control of your production, we would like to have information about the manufacturing process of your product in terms of workflow, quality controls, manufacturing process validation, and also quality control for batch release. Additionally to that, we will also like to have proof of control of your critical suppliers, ideally to have a copy of your quality assurance agreement that you have with those critical suppliers. And finally, we would like to see that you have a process in place which controls the design and development changes on your product. In the technical documentation, we will need first to have a description of the device. We will need to understand what is the product, to understand the product which will be put on the market, and also to understand what are the configuration and accessories which are required in order to make the device work. So we'll need to identify first the identification of the device. It means the name, also the reference number, and I really underline that we need a reference number. I've currently reviewed some technical documentation, and in fact I've seen that the device has no reference number, so this is not possible. We will also understand what is detected. It means that, well, for a nucleic acid test, for a molecular test, we will expect to know what is the target, what are the target proteins, or what is the target region of the virus which is detected by this the in vitro diagnostic test. We will also try to understand what is the intended use. Is it a test for screening? Is it aid for diagnosis? What are the kind of population which is intended to be on which the test will be used? Is it a population which already have symptoms? Is it an asymptomatic population? So this has really clearly to be identified. Additionally, I've also often seen the fact that in this first description of the device, we don't have an idea if the test is qualitative or quantitative. This is one of the first information that we should have. Does the test give a qualitative answer, meaning yes or no, or are we talking about the quantitative measurement of the biomarker? Additionally to that, of course, there's this question of the specimen. What kind of specimen do we use? Are we using nasopharyngeal swabs? Are we using sputum? Are we using blood? Are we using urine? Well, clearly, and here is something that I really like to underline. When we make a claim of specimen types that can be used, we need to prove, we have to have the proof that the test works with all those specimens. Meaning that if my test, I come with a claim saying that my test can be used with blood, serum, and plasma, I need to have the proof in my technical documentation that indeed, while measuring an analyte in blood or serum or plasma, I will get the same identical answer and result. The other question was, what is the tested population? Is it adult, children? We have to have this information. Who is the user? Again, as you've seen, depending on the user, you may have some different classification of the test, if the test is for self-test or if the test is intended for health professional. What is the principle of action? We have to have a description on how the test is working. is it an automated assay or not? Of course, if it's an automated assay, you have to have a description on what is the machine on which the test can be used. And of course, I'm coming back to claims. For each of those machine on which the test can be used, you have to have the proofs and the validation of the use of the test on those specific machines. Also, the question is on the variants. Do you have different variants? As I told you, I've seen tests for their identification, they didn't have a reference number, but they have different variants. So, clearly we have to identify how many variants you will put on the market and to be able to make the difference between all those variants. If the test is coming with a software, of course, and this is very important, you have to give the description and to explain how a description of this software. And again, very important to have the validation and verification of this software as well. If the test contains some substances or tissue of human origin, you should give information on that, although this may not be so applicable to COVID tests. Finally, if you have control material, you have to give a description of those control material to explain from where they are coming, what is expected to have. Those control, do they have a quantitative value, or it's only have a qualitative value? And of course, last but not least, you also need to make a list of accessories or products which are not provided to the device, but they are required in order to make the device work. For instance, to give you an example, if you have an ELISA test, clearly with the COVID, with the kit, ELISA kit, you will not provide the ELISA reader, but you will expect that the lab who is using your test will have this ELISA reader. The same for molecular test. If it's a RT-PCR test, you clearly will expect that the lab which will use your kit will have a PCR machine. From a regulatory perspective, when assessing a technical documentation for a test and also for COVID test, we are also expecting to see that the classification of the device was properly done. So we will expect to have a justification of the classification of the device. And also, it is appreciable to see that the legal manufacturer holds a list of countries where the device it's intended to be put on the market. And additionally to that, we will expect as well to have in this list, as I told you at the beginning of my presentation, that although we have this common market, which is the European market, because of the implementation and transposition of the directive into the national laws, we may have some country-specific requirements. So what I will expect here in this list is to also have the information on, depending on the countries where you would like, the legal manufacturer would like to put its product on the market, is to also have the information what are those country-specific requirements for the registration of the product. And of course, in the case of the COVID test, and as I showed you before, you have some countries where you really have COVID-specific requirements. Would be probably of interest to have a list of those country-specific requirements, because additionally to the fact that will be needed in order to put your product onto the market in those countries, it may also happen that those requirements, country-specific requirements related to COVID, will change or will have an evolution in the next months. So it's also a way for you to make sure that you update the regulatory requirements, and you are aware of changes in the regulatory requirements. So when we put a test on the market, we need to be compliant with the essential requirements as given in the Annex 1 of the in-vitro diagnostic directive. Here at Medide, when we are assessing the technical documentation, in fact, it's exactly what we are doing. We are trying to see that the legal manufacturer holds proof of its compliance with those essential requirements. So the way the legal manufacturer is performing that is that he has what we are calling a ER checklist. It's a checklist of all those requirements. They are more than probably 10. You have, as a legal manufacturer, to go through all those requirements. Those are requirements related to the quality management system, to the development and design of the device, also to the risk management, performance of the device, use of the device, and so on. And you have to show the applicability of each of those essential requirements to your product. So if one of those essential requirements is not applicable to your product, you have to have a justification for it. You cannot just write down not applicable. You have to explain why. Typically, to give you an example, a lot of those essential requirements are related to self-tests. Well, if your test is not a self-test, then you will put non-applicable, and your justification will be the test is intended to be used by health professional, is not a self-test. Additionally to that, you have to show what are the standards that you apply in order to meet the essential requirements. Here, what is advisable and it's commonly used is to use a harmonized standard. If you are European harmonized standards, if you are not using harmonized standards, you have to make a justification. Why do you think that other standards or other way of proving the essential requirement is better than using the than using the harmonized standards? You also have to show the method, to explain what method you use to demonstrate conformity. So you may, for instance, if you have we are talking about an essential requirements which is related to the performance of the test during the whole life cycle of the product. What we can see, we put here would be, the verification and validation activities, analytical performance activities, and including the stability studies, and also probably the risk management file to show that the risks of the device through transport, for instance, was assessed. And lastly but not least, you have to have a list for each essential requirements which you are assessing. In fact, you have to show and to have a proof, a document proof. So the reference to a document which proves that indeed you demonstrate conformity to this particular essential requirement. One major part of the essential requirements checklist is also to check that the risk is managed. So the risk management. There is a harmonized standard which is called ISO 1409071, which gives you information on how to apply the risk management to medical devices. Well, first of all, what is expected to be seen in the technical documentation is to have a risk management plan where all your risk management activities are planned. We need to have a risk analysis where, in fact, you identify risks. It's of particular importance to identify risks which are related to the false negative and positive results. For COVID, it's important not only for patient but also for the community health. So this is very important to analyze what are the risks related to false negative and positive results. Also to check to identify what are the risks associated with the product instability or to the use of the product. Typically, if the test is very difficult to be used, it may generate erroneous results, which may have an influence on the management of a patient. So this is very important to be assessed. Each risk which is identified during the risk analysis it's need to be assessed, meaning that we need a scoring, which most of the time is based on the occurrence of risks, the severity of the risk, and also the detection of the risk. We normally have a threshold. Based on this scoring, we will fix a threshold. So the legal manufacturer has to fix a threshold, and risk needs to be mitigated. And of course, we need to have the documentation which proves the mitigation of those risks. At the end, we also expect to have a risk management report where we have a summary of all the risks which were identified and where mitigation were required, and also an overall conclusion where it says that all the risks identified are decreased as far as possible, and also that there is a risk-benefit ratio is positive for the use of the device. So the legal manufacturer will need to hold the proof of the control of the device design. For this, we'll need to have some data on the device specification, also a list of ingredients, and also, for instance, we will expect here to have some information on the for example, if the test is a molecular test, we will expect here to have the sequence for the primers and for the probes, or the epitopes which are used for the serologic tests. We will also need to have information about the controls which are provided with the device. Here, I would like to underline the fact that for the molecular tests, there are several indications in the current guidelines in Europe of the need of having an internal control on those molecular tests. And this is not the case for all the molecular tests which are currently on the market. We will also need to have information on the specimen type, how the collection of the specimen is conducted, is there a specific material for that, how the specimen processing is done, and also we'll need to have information about the safe disposal of the device once it's used. And finally, all those specifications, in fact, we need to have a plan, in fact, a verification and validation plan for this specific device. So the performance of a device shall be monitored and proven by the legal manufacturer. So, in fact, the legal manufacturer should have evidence of the analytical and clinical performance of his device. He needs to use harmonized European standards. I've put here the document, the European document, the most updated list of harmonized standards which apply to in vitro diagnostic devices. Of interest for all the legal manufacturer, now the harmonized standards which have to be used for in vitro diagnostic devices are currently made freely available by national standardization bodies such as BSI or AFNOR in France. So do not hesitate to go on those websites and search for the harmonized standards, read them and see if they apply to your device or not. Additionally to those harmonized standards, which I told you they are freely available for the moment, you also have guidelines which are quite convenient and very useful when developing in vitro diagnostic device, and this of course applies to COVID test as well. Here I put a list of the guidelines that you may use. Those are general guidelines which apply to all in vitro diagnostic devices. You have those from the International Medical Device Regulators Forum, which IMDRF, formerly called Global Harmonization Task Force. You have a lot of information through those guidelines, very useful information. You also have, of course, the MedDev guidelines, which are a European working group made from national authorities, notified body, manufacturer, under the responsibility of the European Commission. And also you can also find some guidelines which are very interesting to use. which are the one from the World Health Organization for their product. In fact, they implement some guidelines for their pre-qualification program, but those guidelines, in fact, you can use them through the validation of your any in vitro diagnostic devices. And finally, we have this Clinical and Laboratory Standard Institute, which has what is called the EP series. So those CLSI promotes the development and use of voluntary laboratory consensus standards and guidelines within the health care community. So those guidelines, I found them particularly interesting because contrary to the harmonized standards, which give you a goal, or let's say to reach, those CLSI guidelines, it really gives you like the recipe on how to conduct your each specific validation verification activity. So it's quite interesting on setting on, it gives you hints, in fact, on how to set up your performance studies. So additionally to harmonized European standards, additionally to the in vitro diagnostic guidelines, general guidelines, we also have specific guidelines for COVID. There are some of them which I found particularly interesting to use. I put here at the top of the list the ones for the World Health Organization. You see here that you have one called PQDX347, which are for nucleic acids, and the PQDX352, which is for serologic tests. So those guidelines, in fact, were provided by the World Health Organization, and that were made to provide guidance to interested United Nations Procurement Agency, but also to national regulatory authorities throughout the world, not only in Europe, on the quality, safety, and performance of in vitro diagnostic tests for diseases which are declared as a public health emergency of international concern. Of course, COVID is a part of this program, and now there are those specific guidelines which may be applied throughout the development program of your product, but also when assessing the quality of the technical documentation. Additionally to that, we also have some interesting guidelines made by FIND. So FIND, it's the Foundation for Innovative New Diagnostic. It's a non-governmental organization which works together with the World Health Organization. So the FIND conducted independent evaluation of in vitro diagnostic devices with Geneva Hospital in Switzerland. They are providing, in fact, the protocol that they have used during those independent evaluation. I would like to say that the protocol is quite short, on how it can be used if you, as a legal manufacturer, would like to make an independent performance essay by another, by a university, hospital, for instance. You may try to use the same protocol. This protocol, in fact, gives you a kind of scoring criteria based on limit of detection, regulatory status, type of organization, quality management system, and so on. Finally, also the European Commission has issued its own guideline, which are for in vitro diagnostic tests designed for COVID-19. Those, let's say, they are giving some hints on what would be the requirements in terms of analytical performance and also clinical performance. I strongly encourage you to go through those guidelines and when assessing, not only when assessing your technical documentation, but also prior to the assessing your technical documentation, meaning that when you implement your analytical and clinical performance activities, you may get in the information required and how to set those activities based on those guidelines. And lastly, when we are talking about harmonized standards, also general guidelines for IVD, specific guidelines for IVD, which were coming either from the World Health Organization or from the European Committee. But additionally to that, what you have seen with COVID is that we also have some specific guidelines at the country level. So you may find this, you may check it during the either during your technical documentation assessment or during the time you are developing your own test, if you are currently developing it, is to check if in the countries that you are targeting, if they have specific requirements, either in terms of sensitivity, specificity, clinical sensitivity, specificity, or LOD, or in the target on the for instance, for molecular tests, if there are specific targets, viral targets which are requested, do you need to have one target, it's enough, or do you need two viral targets, do you need an internal control, or so on. The performance of a test is given by its analytical and clinical performance. The analytical performance studies may have different aspects. We can check the accuracy of the test in terms of repeatability, reproducibility, recovery. Its analytical sensitivity, it means what is the working range of analyte that can be measured, what is the concentration in terms of linear range, what are the limits of blunt, of detection, and quantitation. We can also talk about the analytical specificity of the device, how specific is the device to recognize a specific analyte. And also we can have appropriate criteria for specimen type, collection of the specimen, and also handling. And finally, we are also talking about the stability of the device. When performing analytical performance studies, we have to be sure that we are using specimens, so samples, in which we know the true value of the analyte, meaning that we have to be sure about the characterization of the samples which are used during the analytical performance studies. When we are talking about the characterization of the samples that are used during the analytical performance study, it means that we need to have a method which assure us about the trueness of the analyte in those specimens. And normally to do that, we need a reference method. This reference method is normally a state-of-the-art method, or the method which is commonly recognized as being able to measure the analyte that our test is intended to detect. One of the first steps when testing the analytical performance of the device is to be able to show that independently of the specimen, sample that is used, sample type which is used, we will obtain the same measurement of the analyte or of the biomarker. For this, what is typically done is that we are doing a matrix equivalency test on which in parallel we are testing the different specimen type that we are claiming, and we are showing that independently of the matrix that is used, we will obtain the same measurement of the analyte or of the biomarker. Additionally to that, we need to show that our samples are stable independently of the matrix used during sample collection, processing, and also storage. Ideally, to give you an example, if we are using blood, plasma, and serum, ideally will be to have from the same donor plasma, blood, and serum, and show that independently of those matrices, we will obtain the same measurement of the analyte. Usually what is done and what is recommended by the World Health Organization is to use for each specimen type a minimum of at least 10 specimens with concentration around the limit, lower limit of detection. Also, the analytical performance of the device really depends on the repeatability and reproducibility of the result. It means that we have to make sure that when using our device, every time we will obtain the same measurement. So we have the repeatability, which is to show that within the same run, we will obtain the same results, the same measurement. Also, that depending on the day of independently of the day of the run or the operator using the device or the instrument which is used, we will obtain the same measurement of the analyte. Both, in fact, the repeatability and reproducibility study should include specimen in the whole range of the analyte, meaning that we have to use samples which are negative to samples near the LOD, and finally the positive samples. We will also need to determine what is the lower limit of detection of our device. And this, it should be shown for all the matrices that are claimed, so all the specimen types that are claimed. Of course, for the LOD detection, we may use spike samples. Spike samples means that they are samples which are considered as being negative, and we spike them with the proper analyte at concentration which are known. For instance, for molecular tests, we can use negative samples which are spiked with known concentration of the inactivated virus or cell culture-derived viruses. Normally, at the level of the LOD, we should have a minimum of 95% of the samples which are positive. If in fact we see during our experiments that we obtain 100% of tests of the results which are positive, it means that probably the LOD is lower than what was expected, and we have to perform again activity studies for the detection of the LOD. As I told you, we need to determine the LOD for each claimed specimen type, and normally the LOD is established using at least 20 replicates, which are spanning the tentative LOD. So what is expected to be the LOD concentration. We also need to check what if we have substances which may have an influence on the result obtained with our device. This is called interference, and we may have endogenous substances, like for instance when using nasal samples, we can have mucins, or if it's in the blood, we can have cholesterol, triglycerides, which may interfere with the proper measurement of the analyte. So normally, what we are doing here is that we are using for each endogenous and exogenous specimen, spiked and unspiked samples with the analyte at concentration of the analyte around LOD. The substance which is a potential interferent, either endogenous or exogenous, should normally be tested at very high concentration to be sure that even at high concentration it will not interfere with the result obtained on the measurement of our specific analyte. Again, normally the interference should be measured for all the specimens type which are claimed. However, if we show that there is equivalence between matrices, we can avoid using. We can perform the test only on one matrix, which is considered as being representative of all the other matrices. There is an interesting CLSI guideline which is called the EP07. Interference testing in clinical chemistry, which gives you a very interesting. It gives you the design of the interference tests. It also gives you some concentration on the endogenous, exogenous substances. So it gives you hints on what are the endogenous and exogenous substances that need to be tested. And also, very important, you can use specifically for COVID the World Health Organization guidelines, which I've mentioned in one of my previous slides. In fact, in those guidelines, you find a list of all the endogenous and exogenous substances that are to be tested in order to avoid or to test, if not interference, with COVID tests, either for molecular tests but also for serological tests. We also have to check during the analytical performance studies that our device will not cross-react, will not recognize other analytes which may resemble to the analyte which is detected by our device. So in case of the molecular tests, what it should be done is to be checked that our test will not recognize other viruses which resemble COVID-19, such as MERS, influenza, coronavirus, other coronaviruses, and so on. This can be done in the first step by the in silico alignment of all those organisms. And of course, for some of them where there's some homology seen, specifically for the region which is targeted by the molecular test, it should be performed, wet lab testing should be performed. For the serology testing, it's also possible that other antibodies which are raised by our organism in contact with other similar viruses or infection may be unspecifically recognized by our test. So here again, we should test that there's no such cross-reactivity. For those of you who ask the question, so what do I'm supposed to know? What are the viruses or bacteria which I have to test? Well, there is again the World Health Organization guideline that I've mentioned previously. Give you a list, as for the interference studies, it gives you a list of the cross-reactants which need to be tested for tests which are designed for COVID-19 detection. As I said at the beginning when discussing the analytical performance, some analytical performance studies may differ from because of the type of the test. Clearly for serological test or molecular test, we have some specific activities that should be underlined. Here in the case of molecular test, clearly we have to be clear on the target sequence. What are the sequences which are targeted by the primer and probes? We need to have a rationale on the design of those and why targeting the specific region. And of course, we need to have some alignment between the sequence of our primers and probes with the sequence of different viral strains which may be comparable to COVID-19. So I'm talking about MERS, SARS, and so on. A major drawback, in fact, for serological tests is the hook effect, and clearly this has to be assessed in the case of serological tests. This hook effect, in fact, happens when you have when an important concentration of the analyte is present in the sample. While doing that, in fact, it washes away the result, and as it's presented here, is that with a very high concentration of analyte, you will arrive to have a signal which is lower than the signal that we may have with a lower concentration of the analyte. And in some cases, of course, it brings you to have a false negative result. So as false negative results is an issue for COVID-19 detection, this clearly has to be, the hook effect should be investigated before putting a product on a device for COVID-19 detection on the market. One of the biggest questions when putting out the test on the market is how stable it is. How long can I use? So shelf life means how long I can use the test. So of course, we are here with the COVID, which is a situation, unexpected situation. Most of the manufacturers who develop tests for COVID do not have the feedback on the stability, real-time feedback on the stability of their test. So what it's allowed to do, and this is clearly described in the harmonized standard related to stability, which is the ISO 23640, in case where real-time shelf life is not available, we can use accelerated studies and extrapolate to this data to the shelf life of the device. Of course, this is acceptable only if we have proof that real-time stability data is currently ongoing. Additionally, for that, most of the manufacturers are developing tests which are to be put on the market not only in one country or on the premises of the manufacturing facility, but it's also to be transported elsewhere in Europe, but not only. So the question is, what are the conditions on which I can transport my test? So here, in fact, every manufacturer, depending on the target countries or target geographical markets, should mimic the extreme condition of temperature, humidity, if the humidity may have an effect on the performance of your test, and also the pressure, and to perform studies to show that when transporting the test in those conditions, this will not have an influence on the performance of your test. Additionally to that, the question is also, what is the stability of the test once it's open? So for this is called the in-use stability. And here we will have studies such as vial stability, vial open vial study. So those open vial studies, if I take an example of lateral flow tests, if the lateral flow test is coming by a package of 20 lateral flow tests and one bottle of the buffer, we may open the vial and see how long the stability, or what the performance of the buffer is kept, it's maintained once we open for the first time the vial containing the buffer. In the other round, and also for lateral flow test, we can also ask the question, well, what happened if we open the pouches? Normally lateral flow pouches, they are coming individually in aluminum pouches, because lateral flows are known as being sensitive to humidity. So one other question would be, what happened if the seal is open? How long can I keep the test before performing it? So this kind of studies are done during these stability activities. Up to now, we have discussed the intrinsic performance of the device, which is called the analytical performance. We have discussed about its stability. We discussed about the repeatability, reproducibility, limits of detection, and so on. Now we would like to know how the device performs when in the real life, let's say, when used as it should be used, but depending on its intended use. So this performance, real life performance of the test, is called clinical performance. Normally, when conducting clinical performance studies, we should use specimens from all sections of the population for which the claim is made in the instruction for use of the device. Here, for instance, I will expect, if the test is to be intended for a European population, I will expect to see studies which were performed in a European population and not on an Asian population, to give you an example. Also, it is very important that the samples which are used, we are talking here about samples which are used retrospectively. Ideally, of course, it will be to conduct retrospective samples, but most of the time, the first studies which are conducted are used on retrospective samples. So now, when we are talking about the specimens, as I told you at the beginning of my part on the analytical performance, it is very important that the samples which are used during the analytical performance need to be clearly characterized by a reference method. The same is in a way applied here. When we are using sample, we need to have a predicate, we need to have another method which is commonly recognized by the medical in the medical field. If we are talking about COVID, clearly for the moment, the only methods which are recognized are the PCR method, molecular method, not serology. So in fact, the samples that we are using need to be characterized when described as being COVID positive or COVID negative, of course by the symptoms, but also by another recognized method, and in this case, molecular method. Normally, when clinical performance is done, it has to be done for each claimed specimen again, and I really underline that because I'm seeing it very often in the technical documentation that I'm reviewing. We need to, if you claim that your analyte can be measured by your test in sample of blood, serum, and plasma, we are expecting to see data, clinical data from blood, serum, and plasma. Another question which is often asked by our clients is how much sample do I need when conducting clinical performance studies? Well, I have a tendency to say that here normally the legal manufacturer knows better what is needed. Of course, you need to check the strength of your studies to have a statistical power. But by saying that, and to give you more easy hint on how to conduct your studies, well, I think that it's good to go directly on the World Health Organization guidelines, on those documents which I told you about, PQDX 352 for antibodies and 347 for nucleic tests, where it gives you what they are expecting as a minimal clinical performance assay data. So here you see that for molecular tests, they are saying that to calculate the clinical sensitivity, you need at least 50 samples. Ideally would be to be prospective, but this is often not the case. So it's also said that you can use 25 retrospective prospective, and you can also use contrived samples which are around the LOD and a bit higher than LOD. For the clinical specimens, for molecular tests, a minimum of 100 negative samples collected from symptomatic patients is requested. And you can see that the serology, those numbers are hugely increased. We can see here that for the clinical sensitivity, we are talking about 200 specimens. And for the clinical specificity, we are talking about more than 1,000 specimens. So as a conclusion of my presentation, I would say that the registration of COVID tests, which are intended for self-test, need the involvement of the notified body. This is not the case for the health professional use tests, which are going to the European market by a notification to competent authorities, following a conformity assessment procedure, which is conducted by the manufacturer itself, who also declare that the essential compliance to the essential requirements as per Annex 1 of the IVDD. Do not forget their COVID-specific, country-specific limitations. So before going into a country with your product, check that what are the specific limitations in that particular country. Also, I strongly encourage you to check the guidelines, which are the international and also national guidelines, which were issued on COVID when preparing the documentation and also when developing the test in order to prepare your clinical and analytical performance studies. And finally, concerning the humanitarian procedure, do not forget that this is a country-specific application, so you have limitation in time and also restriction on selling this product in each specific country. Thank you very much for your attention.